In this tutorial, I would like to share our experience of what we learned from the past several years on the single-cell proteomics. Single-cell biology is one of the top 10 technology and science area EMSL will pursue in the next 10 years. And we have invested a lot in the directions in the past several years. And we also and go ahead. Okay. So in this tutorial, I will give some time to give a basic of the near single-cell proteomics, and then I will introduce several cases how we working with the user, working with the collaborator, use the EMSL facility to do the near single-cell proteomic researches. I have some short video to show how we process the sample use nanopod after we receive the sample from the users. I left some time for discussion and Q&A. On the basic of single-cell proteomics, first I will introduce the scope of single-cell or near single-cell proteomics, and give an introduction of general considerations, how we prepare the samples, and also how we run these samples use LC-MS. I think I don't have to emphasize, highlight too much here, why we have interest in the single-cell proteomics analysis. And if we look at the real-world samples, no matter it's human samples or plant samples or microbial communities, they are very beautiful. They are not just single population. They are not just single type or single cell state. They are a mix of different cell types and mix of different cell states actually, and they are well organized, working together to perform the biological functions. So single-cell analysis can help us to have an in-depth understanding the cell functions, also understand the variable response to environmental perturbation or stimulations. Our very important part is we can capture rare cells. We usually cannot see them in bulk analysis, but they play very important biological functions. This is the scale of current proteomics researches. And traditionally, usually the traditional workflow usually working with the biological sample have protein amount over 1 microgram. And usually, we require 1 million cells, at least 100,000 cells, have about 5 microgram of the proteins. Usually the tissues, plasma, biopsy, or cell cultures. And there's a more interesting part is get into single cell or near single cell, small number of cells. We define the near single cell proteome is the sample contain the total protein amount below 1 microgram in nanogram amount of proteins. Usually it's salted cells, either enriched cell population usually, or use LCM cut tissues and embryos like the introduction of Peter today, or even exosomes or cultural secretomes, or even circulating tumor cells. There's some commonly used term of the near and single-cell proteomics. Usually we see from literature. The first one is single-cell proteomics, or SC proteomics, is usually referred to proteomics analysis of a single cell. Another usually used the term is single cell type proteomics or cell type specific proteomics. It's not working on single cell, actually it's working on the pooled cell populations like 100 cell or even 1,000 cell, but they are enriched the same cell type or same populations. So we can get the different information from different cell type. Another one is usually use spatially resolved proteomics, or the newly term is proteome imaging, and it's use proteomics technology to analyze the tissue pieces from usually from laser capital microdissection isolated tissue part, and usually from different locations. We can see the protein abundance at the different locations. Maybe they're not from same populations, but they're from different locations. When we consider to do the single cell or near single cell proteomics, we need to also consider what's the species of the samples. So here is a very good table to summarize the four commonly analyzed species, like bacteria, like E. coli and yeast, and plant. Also human cells. If we look at the volume of them, share my data point. If we look at the volume, it's E. coli is very small. It's only usually it's 1/5 more, and because it's so small, usually if you look at the protein copy numbers, it's very low. It's usually less than 1 atmole. For the yeast, is a little bit larger, but still very small. So the plant human cells usually large enough. Usually, if you look at the copy numbers, plant have very high copy numbers. And human also pretty good is usually is less than 1/5 more. It's 108 more or less. So if we considering the challenge of in single cell proteomics is the human samples or mammalian sample is easier, and the plant cell is also easier. And but the yeast, the E. coli is really challenging to do the single cell proteomics. So when we do this, if you look at the table here, I would like to say that currently we cannot do bacteria and yeast, but we can do plant and mammalian for single cell proteomics. For single cell type proteomics or spatially resolved proteomics, you can do all of them. If we look at the workflow of current bottom-up proteomics, you usually have 3 major steps. So the first step, we get the cell or tissue. We need to lyse the cell to release the protein and release protein into the solution. Then we do multi-step denaturation and alkylation, and finally digest the protein into small peptide. The reason we need to digest them because they are well separated and have better ionization efficiency and are easy to be identified by MSMS. Usually we use liquid chromatography. Also we can use capillary electrophoresis. The LC separation can reduce the spectral complexity, and the also reduce the separation also can increase the ionization efficiency by reduce the ionization separation. Finally, after get all the data set, we usually use the database search tools to matching the MSMS spectrum to the database predict spectrum to identify the protein, and then iterate the protein intensity to assembling into protein intensity to quantify the protein abundance in the samples. So for the sample preparation, usually contain the cell isolation step and the cell lysis, protein extraction, alkylation, digestion, and sometimes we do some labeling. The cell isolation usually is a key step for single cell or near single cell analysis. So most commonly used tools is flow cytometry. So we have one in EMSL. So we like it because it's very powerful. It can isolate individual cells based on size, shape, and even you can use antibody or other fluorescent labeling to select the cell population you want to analyze. It's very high throughput. We believe it goes for bacterial fungi or mammalian cells. And for the plant, we need 2 actual steps to remove cell wall to generate the protoplastas. In addition to flow cytometry, we also have another technology here is CellenONE. Unlike the flow cytometry use the laser to identify the single cell, CellenONE use another approach, is use a imaging approach. It use you take the image of each cell and use the machine vision, machine learning to identify the cell, and then moving the dispenser to where to dispense the cell out. So compared with the flow cytometry, it's a relatively low throughput. It can do 10 to 100 cells per minute, but still good enough for single cell proteomics because you are not going to run those single cells. We like it because it's compared with the flow cytometry, it only requires very few cells. If you have 1,000 cells, CellenONE can precise sorting them without lose a lot of your precious samples. General considerations, if we working with ours or send samples, prepare samples in your lab, at first we have to do dissociate cells if we use flow cytometry or CellenONE. And in terms of the fixed cell or non-fixed cells, we always find out unfixed cell always give a best protein recovery. Because usually for fixed cell, the most common use is the PFA or formalin fixed cell. All the protein is cross-linked together. So it's really hard to decross-link the protein and recover them for proteome analysis. We will give the worst data. It still gives something, but the quality is not very good. We also try use a methanol fixed cell. It's have moderate recovery, and we see it during the fixation. We see it lose some small proteins and hydrophilic proteins. And for the phenotype labeling, we do not see any problem. So it's working. We are working on antibody labeled cells. We're also working on other fluorescent label cells like the calcium, cell viability dye, and the dipay also works. Also, some other memory dyes, we don't see any problem. For plant cells, it's a different story because we have to digest the cell wall to generate the protoplastas. So you can send us the methanol fixed protoplastas, or easy way to do is bring the proprietary tissue. We can generate the protoplasta in the lab and direct to the sorting. In another cell isolation technology we are using a lot is laser capture microdissection. Another name is LMD. It's a laser microdissection. Yeah, different vendors use different name. The one we are using is this one. The LCM is just look like just a microscope. Actually, it integrates high resolution microscopic imaging with laser ablation and injection. So we can isolate the tissue as small as single cell. We can cut a very, very small size of the tissue. Also, it's compatible with antibody labeling or GFP fusion technology. We can use because we have a 4-channel fluorescence to identify the cell of interest. The procedure of the laser capture microdissection is here. Generally, you use a microscope to image the whole tissue sections and find out the cell of interest or area of interest. So you draw a line across this area, and then use the laser the laser will start to cut. Use your drawing, and the last step is the laser will generate pulse. The pulse will inject the sample into the collection device. It's the same video showed by Ryan. So it's just show you how the LCM works. The first step is laser ablation, just isolate the tissue from the, isolate the pixel or box from the tissue. We usually do 1 or twice laser ablation, make sure it's completely isolated from the tissue. And then it applies laser pulse to the center of the tissue or to the edge of the tissue. It will shoot the tissue into the collection device. Usually it works pretty well, but sometimes we will lose the small, if the tissue is too small, because electric charge, it just disappear after laser ablation. For cell lysis protein extraction, we usually use single port protocol because, as Ryan and Peter discussed this morning, so the actual transfer will have more sample rules. We will have any transfer between each steps and no buffer exchange or clean up. So what we do is put the sample inside the wells and do multi-step like extraction, alkylation, and digestion, and remove the surfactant by the acid. So there's because we cannot do purification, we have to use mass-compatible surfactant. So there's a lot of available surfactant can do this. So like refugees from waters, protein marks, from Proteome or Evitrogen from Thermo Fisher. The one we are using a lot now is the DDM. This we found out is a very good surfactant for the single port sample preparation for proteomics. It have a dual function for the single cell proteomics. The first function is can do very efficient cell lysis protein injection. Also, it can increase the sample recovery, and it will not affect your LCMS analysis, because usually if you look at the LC separation profiles, all the peptide come out before 100 minutes. So the DDM usually is a big peak will come out after all the peptide come out. And for the peptide recovery, you can see with the DDM, we always increase the load on the protein identifications. We have a much better recovery for the if DDM is added into the buffer. If we look at the protein recovery, it's much larger. We compare different surfactant. Generally says without DDM, all the protein will lost to the container surfaces. For the alkylation, we do not have we do not see a effect. This step will affect the proteomic analysis, so you can get rid of it, or you can still do it. It's just one extra step, just 30 minutes. The major challenge we see is the protein digestion, because the low digestive kinetics for the single cell or small number of cells. So if we look at if we lyse one cell in 100 microliter, we will reduce the concentration to 1.6 milligram per ml. Typical digestion will usually is 1 milligram per ml. So the solution is you can add high amount of protease, more protein, more trypsin, more lysin C. And another way is the one we are working on is minimize the volume with nanopore technology. So usually we do the 200 nanoliter digestion. So we only add 1 nanogram lysin C and 1 nanogram trypsin. Also, Isobaric labeling is a technology to increase the throughput of proteome analysis. It's a reagent introduced by Thermo Fisher. It's commercially available. The beauty of this reagent is have a lot of different tags. It's the MS1 is completely the same. So, but you can see the star is the heavy labeled table of nitrogen, it will change the location of the heavy labeled element and at a different position in the mass normalized mass reporter. But after HCD or MS2, only report ion is released. So you can see the different report ions can tell you the different protein identification, protein quantification information. The general step of the TMT labeling approach is you do all the sample collection and protein injection, digestion. After digestion, you label each sample with one unique TMT tags. After labeling, you can combine all the labeled samples together into one sample. So you use one sample, you can run multiple samples. If you look at the spectral, all the reporter ion is here. It's usually from 126 to 131. So this reporter ion can tell you the protein abundance different across the different samples. The TMT-based single-cell proteomics is a little bit different from typical TMT approach. It's still do the single cell isolation and do the digestion and do the labeling, but the difference is at 200 cells as a carrier. So because there's more cells, that can help to the identification because it provides more fragment during the MS analysis. But because it uses the carrier protein or booster protein, you have some limitations. Like if we use one channel at both have 100 more materials, if you look at the contamination table here, it will contaminate other channels. You have 7% contamination to 127C and 0.2% to 128C. So here is the data we get from our runs. So we add 100x more sample in the 126 channel. As you can see, even we have 4 channel is blank, we still see a lot of report ions in the 4 channel, and the more report ions in 127C and 128C. It tells us we cannot use these channels. And if we look at the protein abundance, report ion abundance, it tells us this channel and this channel, 127C is completely contaminated, and also 128C also cannot be used. So if we have one channel, we will reduce at least 3 channels in the TMT set. But those still have some channel can be used for single cell analysis, still valuable of this method. For LC-MS, the general consideration of the LC column selection is how we can increase the sensitivity of the proteomics analysis because we cannot amplify the proteins. We all rely on absolute sensitivity of the LC-MS analysis. What we learned in the past 10 years or 20 years is if we want to increase the sensitivity, we have to reduce the column size. Because the electrospray ionization is a concentration sensitive ionization method. So decrease the column size can lead to high concentration of the peptide at the ESI source. It will increase the sensitivity of your peptide measurement. And here you can see we have 4 different size of column from 75 to 15. So at 75, we inject the same amount of sample into the 4 different columns. So most see few signal in the conventional 75 micron column, but we can see a huge amount of signal at the 50 ID column. So usually we use the 50 ID or 30 ID column is for single cell proteomic analysis now. For mass spectrometer, we also need to choose the best mass spectrometer to get the highest sensitivity. So in EMSL, we have 2 mass spectrometer for the single cell proteomics. One is OB-triple Lumos, another one is OB-triple Eclipse. So we find out they have most sensitive and high resolution for single-cell proteomics, and also it can be used both for label-free and isobaric labeling approach. If we look at the design of mass spectrometer, as we know, mass spectrometer is the mass detector. More ions give you more signals. So ions will go through the collection, ion collection capillary, and go through the ion funnel, and selected for MS in the quadrupole here, and trap them in the ion routing multiple, and then use the C-trap to throw into the OB-trap for detection. So that's A2 gain control in the OB-trap. So it's a setting how much total ions we will select to throw into the OB-trap. Another parameter is ion injection time. It controls how long it allowed to accumulate the AGC-defined ions. So because settings, that's different in the setting for single cell. So for the level single cell proteomics, you look at here. For the MS setting, it's almost the same. But for the MS2 setting, for both proteomics, we use the lower resolution over AGC and lower IT time because we want to get coverage. We want to do more MS. But for single cell proteomics, we usually use the high MS2 resolution to do the MS2 scanning. Also, we usually use high AGC level. The most important parameter for the level-free single-cell proteomics is the ion injection time. So we use the 250 millisecond or 500 millisecond to allow the mass spectrometer to accumulate more ions from a single cell for detection. For isobaric labeling, it's a different story because we have a carrier channel or boosting channel. What we found out is if we use the similar setting, we have more trouble because if you have one channel have 100x more ion, more proteins. If you do AGC trapping, all the ions actually not from your single cell, it's from the carrier protein or booster protein. So to increase the detection of the single cell ions, we have to increase the AGC setting. So we usually use the very high AGC setting for the isobaric single cell proteomics. So here is you can see if we use 5E4, the signal is really low, and if we increase over 5E5, we can get very good signals for single cells. And if we look at the coefficient variation, the CV, and also tell us we have to select very high AGC level over 5E5 to get the better CV. This is a typical setting for the iso labeling based single cell proteomics. We use the 5E5 or 1E6 for MS2 AGC. And we use, sometimes we use 250, sometimes we use 500 millisecond, depending on the cell type. Now, I finished the basic of single cell proteomics. I will move to the case studies. Is several examples how we collaborate with other institutes or other users to use the tools developed in EMSL to advance their researches. The first example is single cell proteomics of inner hair cell development. So this is a study in collaboration with OHSU. The PI in OHSU is have interest to study the inner hair cells. Is the mechanical transduction cell. Is the cell responsible to transit the sounds into the electrical signal so we can hear the voice. The problem of the study is the hair cell is it cannot be regenerated. So if you lose the hair cell, you lose the hearing. And also for biomedical studies, it's very challenging to study it because in 1 year only usually has 700 in the hair cells. You have to select very sensitive detection method to do the measurement. So what we do, the study we do is we want to look at how the supporting cell slowly differentiated into the hair cell. It's a hair cell function cells. So we send the chip to our collaborators. So they use their photometry, and we do some back and forth to do a training, how to train them how to sort the single cell into our nanowire chips. So they use the fluorescence is FM143. It's very interesting. The only hair cell can be labeled with this fluorescent dye, and the supporting cell is usually the non-fluorescent. So we sort both the MF133 low and the high fluorescent one, and put them into nanowires. So the collaborator then send us the chip, and we receive them and do the same prep in here. So we didn't get very high coverage, but still tell a lot of good stories. Use data analysis like a cell tree is a trajectory analysis tools. We can build up a pseudo time trajectory to show how rank the single cells is rank single cell based on pseudo time, based on protein abundance. Some protein markers differentially expressed in hair cell and supporting cells. As you can see, actually, it also tells us the dye is not specific enough. We still see some cell is a hair cell, but they have no fluorescence, like the top one, and some cell have no fluorescence, but they have high fluorescence, but they are supporting cells. Very interesting, we found that one protein is responsible for the actin release during the hair cell development. So we also do some validation, use immunohistochemistry staining. It's also validate our single cell proteomics platform is precise enough to discover the protein markers. In the second case study is not single cells, is single-Islet. For these studies, we also it's also different because the collaborator do all the tissue sections and also do all the tissue treatment in their lab. So usually for the tissue that we have to go through many different steps. For the fresh frozen tissue, usually the first step is denaturation, use ethanol, and then use water to rehydrate the tissue for the H&E staining. After the H&E staining, usually we do we dry the tissue, dehydrate the tissue, use the ethanol steps. The collaborator will do all the LCM first, so they will find out all the single-Islet and use the laser to cut them, and they will send us the tube. So after we receive the tube, we will do the manual transfer because the well is very small. We have to find a way to put these small pieces of tissue into our nanowires. What we do is we have tweezers, it's ultra fine tip tweezers, and we use a stereo microscope, and we will find out each Islet and manually pick each Islet from the cap and to the nanowires. So we do this a lot. So we cannot guarantee 100% successful rate, but still we can get maybe 90% successful rate. actually depend on the tissue size. We can do pretty well if the single-Islet usually have a 100 by 100 size, micron size. We also try to do 50, still works. This is some result of single-Islet proteomics. You can see it's pretty reproducible for the 9 single-Islet from the healthy control, healthy donors. Here is we analyze the 9 islet from healthy donors and 9 islet from type 1 diabetes patient. As you can see, we can clearly see the insulin is present in the healthy donor, but it's almost disappear in type 1 diabetes patient. In the third case is a different way to do the tissue samples. So we can start from the plant fruit or plant leaf. So we can do this. The collaborator will collect all the tissue or organic organ from the plant and send to us. So we have the capability in EMSL to do the tissue sectioning. After the tissue sectioning, we go through all the fixation and also dehydration steps. So we will directly use the laser capital microdissection in lab to directly cut the tissue into the nanopod device and run them through LC-MS. This is a case study use tomato fruit. So we just use this one as a technology development demonstration. So we try to see if the LCM is good enough to purify the cell types. So we isolate 3 different cell types from the tomato fruit. The first cell type is outer epidermis, and it's only one layer of the cells on the outside of the tomato fruit. We usually isolate about 100 cells in each samples. Another cell type we are working on, isolate is Collinchima is the cell between outer epidermis and parenchyma cell here. So we isolate 3 different cell type and run them through our pipeline. So here is the quick PCA analysis to show their protein expression is totally different. You can see the PCA spread is really well, and also the PC2 also well spread all those 3 cell types. Finally, we have a video tutorial to show after we receive the sample from cell isolation in the lab or from a collaborator or from a user, how we process the sample use our nanopod technology.
Hello, my name is Ying Zhu. I'm a research scientist at EMSL. Today, I will show you our single-cell proteomics workflow using nanopod technology.
Okay, finally, I have some suggestion. If you want to know more the nanopore technology, Ran Kelly have a very good review here. It's just published on MCP. You can, I believe you can download, everyone can download now. Another one is published 2 years ago, also have some basic of the technology development. The original paper of the nanopore is this here. The first one is the first paper for the development of nanopore. The second one is how we coupling with LCM. The last one is how we improve the Isobaric labeling by the single-cell proteomics.